Good morning, all my dear students. So this is your educator Sindhuja over here. So today we will be discussing about chapter 6 life processes of your class 10th chapter. Inside that I am going to teach you about blood physiology. Okay, a small part of that particular topic. First and foremost, do you all know what is blood? Blood is a special type of fluid connective tissue. It is derived from mesoderm. Okay, so it has got mesodermal origin. Now, some extra tidbit facts, extra things regarding this particular blood. That is, you all might be knowing there's a branch of science which deals with the study of blood, blood forming tissues, and the disorders associated with them. That particular branch is known as hematology. Now, the word heme inside hematology means blood, and logos means study. So, like that, this word was formed, hematology. Okay. That is regarding blood. Then we have to know about the properties of blood. What are the properties of blood? Properties means in terms of color, then mass, pH, then taste, temperature, viscosity, volume. All these things come under properties. So color, most of you might be knowing, it is bright red in arteries and dark red in veins. Okay. Then mass is almost, it occupies 8% of the body mass. pH means it is slightly alkaline. That is of the range 7.35 to 7.45. The next thing is regarding taste. It is salty in taste. Temperature, it is 38 degrees Celsius, and in terms of Fahrenheit, it is 100.4 degree Fahrenheit. Okay. The next property is about viscosity. It is 3 to 4 times more viscous than water, and volume is 5 to 6 liters. So these are the properties of blood. So when I taught you about properties, it means properties has got certain parameters, like just now I told about color, then temperature, then about pH, then what else we discussed, like viscosity, volume, etc. That is regarding that particular properties of blood. Next comes the part that is composition of blood. What all things make up blood? You all might be knowing, if you take in a test tube blood, the topmost portion will be plasma, which is a slightly straw colored in nature. Below there will be centrifuged blood or normal blood, where you can find red blood cells, white blood cells, platelets, etc. That means the composition is as simple as that. That is, it comprises of plasma as well as formed elements like WBC, RBC and platelets. That is about composition of blood. Now let's discuss about blood, how it is classified. Like blood has got plasma, I told you a few minutes back, and cellular elements. Plasma comprises about 55% and cellular elements comprise about 45%. Inside cellular elements comes the erythrocytes or RBCs, WBCs or leukocytes, and platelets or thrombocytes. So these are the category means in composition whatever I taught with exact percentage I was describing in little bit in detail. That was regarding this particular thing. Then let's discuss what is plasma all about. Though I mentioned plasma is a yellow colored liquid component of a blood, what is the function of plasma? It holds the cellular elements of blood in suspension in its place. That is the function of plasma. So here I mentioned you about the plasma's color. That is a yellow colored liquid, and then it's a yellow colored liquid component of a blood. What does it do? It allows the formed elements to be in its position. That is regarding plasma. Okay. The next one is regarding constituent of plasma. What all things plasma is made up of? See, plasma has got 91.5% water content. Proteins. Proteins means 7% of its composition is proteins. Proteins means albumins, globulins, fibrinogen. All these things make up the proteins. Then of course you find trace amounts. Chota wala 1.5%. Other solutes, like it could be electrolytic ions. Then what else? Gases. Then nutrients. Then waste products, etc. In electrolytic ions, it means Na+, K+, Ca2+, Mg2+, etc. etc. Gases, oxygen, carbon dioxide, nitrogen. Then nutrients means glucose, amino acids, fatty acids. And then comes last but not the least waste products. Urea, uric acid, ammonia and creatinine. So that is regarding the constituent of plasma. Okay. Hope it is crystal clear for you all. Then actually I have a chart, but since I am not sharing the screen or using any application, that's why I couldn't show it to you. Okay. Next one is regarding functions of plasma. Functions of plasma means it's simple. How you will study? By now you know what are the composition of plasma. Think about those composition and then attach each function related to it. For example, I will make it little more simple. Like I told water is one of the constraints at plasma, right? So what is the function of water? It absorbs, transports and releases heat. So in a sense, what is the function of water inside plasma? That at the end of the day, it will become the function of plasma also. Understood? That means whichever component I mentioned inside plasma, each one of their function is totally known as a function of plasma. Second one about albumins I told. That means albumins have a role in osmotic balance. Okay, that is regarding albumins. Then globulins is there. Globulins is something associated with defense mechanism. Then I told about fibrinogen. It is related to, it's a protein related with the blood clotting. And electrolytic ions you all may be knowing to regulate pH buffering and all. This particular function is maintained. Okay. Now let's look in detail what is RBC. RBC means its technical term is erythrocytes. RBC shape, most of you might be knowing, it is biconcave, circular biconcave and non-nucleated. Okay, no nucleus it has. Size, diameter, it is 7 to 8 micrometer and thickness 2.5 micrometer. Okay. Color, red. Why its color is red? Due to the presence of a pigment known as hemoglobin. Okay. Then regarding count, in adult male it is 5.4 million RBCs per microliter and in adult female it is 4.8 million RBCs per microliter. Lifespan you might be knowing it is 120 days. Okay. So inside RBC you need to know about the shape, size, color and the lifespan. Okay. Then next comes the production of RBC is technically termed as erythropoiesis. Okay. In the case of adult where this production occurs, it occurs in red bone marrow of long bones. Long bones means hip bone, then your breast bone and ribs in the case of adult. But in the case of child, up to 5 years, bone marrow of all the bones has a capability to produce what? RBCs. And in the case of fetus, RBC is produced in liver and spleen. So, I didn't give you a vague explanation about just what is erythropoiesis and one aspect. I told you like 360-degree coverage I told. Like in the case of adult, where is its site of production? Child, where is its site of production? Fetus. Okay. Then associated with erythropoiesis, we need to know the increase and decrease terminology. If ever the RBC gets increased in number, it is known as polycythemia. And if ever it gets decreased in number, whenever RBC shows decrease in number, it is known as erythropenia. Okay. So those are the two extra things. Then next one is functions of RBCs. What might be the functions of RBCs? It is for transport of oxygen from lungs to tissues. Likewise, transport of carbon dioxide from ulta, that is from tissues to lungs. Okay. Now, how much amount of hemoglobin do you think will a normal blood contains? Yes, exactly. It is 13 to 15 gram of hemoglobin per 100 ml of blood. But one RBC can contain about 250 million molecules of hemoglobin. Okay. Then each molecule of hemoglobin has a capability of carrying 4 molecules of oxygen with it. That also you have to remember. Next is regarding white blood cells. White blood cells means shape. First we will discuss shape. It is amoeboid nucleated in shape. Nucleated means it has nucleus. RBC didn't have nucleus, but here nucleus is there. Then about size, it is about 12 to 15 micrometer. The next one is regarding color. In the case of color, it is colorless and translucent. Count 5000 to 1 lakh WBCs per microliter. The lifespan, it is 10 to 13 days. Okay, the same criteria which we discussed in RBC, but here the data will be different. Okay. Next, the production of WBCs is known as leukopoiesis. Okay. In the case of adult, the site of leukopoiesis is liver, spleen, tonsils, bone marrow. Whereas in the case of fetus, it is liver and spleen. Then, the terminology is associated with the production. That is, whenever there is increase in number of WBCs, it is known as leukocytosis. And decrease in number of WBCs is known as leukopenia. Pathological increase in number of WBCs is known as leukemia or blood cancer. Okay. These terms are associated with this thing. Then let's move on to the types of WBCs. So WBCs are of two types, granular and agranular. Granular means the cytoplasm will be of granular texture and internally the granular WBCs are classified into neutrophil, acidophils and basophils. Neutrophils form 62% of WBC, granular WBC, then acidophils 2.3% and basophils 0.4%. So granular thing is over. Next, agranular thing. Inside agranular comes lymphocytes and monocytes. Lymphocytes constitute about 30% of WBC and the next thing, that is monocytes comprise 5.3%. Okay. That is regarding the classification of types of WBC. Then granular WBCs, let's look in detail. In the case of neutrophils, the features, granular WBCs are of three types, neutrophils, eosinophils and basophils. In the case of neutrophils, the features are it has nucleus with 3 to 4 lobes. Then it can stain with neutral dye like hematoxylin. Functions means it can destroy bacteria by phagocytosis, and location where this is produced, where neutrophil is produced, is bone marrow. Then acidophils or eosinophils. It has got nucleus with